No questions? I'm going to go ahead and share my screen and then we can get started then. All right. Hopefully you can see the screen. What we're covering today is we should finish chapter 5. I don't think I'll move ahead. So it means we'll probably get out a little early today, meaning I don't even know if we'll have the second lesson. Also, let me remind you the quiz 1, is due at 11:59 p.m. on Friday. You can start the quiz by midnight tonight, but it has to be done by 11:59 p.m. on Friday. Any questions about what we're doing? Let me talk briefly about something that I think I sent out an email to all of you guys. You can get together either over the internet or over a phone and work together and study together, and that you can then click on this link, the extra credit study sheet.
It's study sheet 2.
I'm sorry? Right, that's the wrong one. Sorry. That one's no longer due. That one's already supposed to be turned in. It's the extra credit study. Week 2, due June 13th for the one that's due week 2. You have to study for at least a half an hour, get together with somebody, and quiz each other. Has to be something that you might be tested on. So if you're just getting together to talk about, what project you can do for your, what is it, the infectious disease project, that wouldn't count because I'm not going to quiz you on that. And then hopefully on the right up here, there's a button for you guys to submit the file. I don't have that because I'm the instructor. I download submissions, but you can upload it. You have to give the full names of everyone in the study group and tell me when you studied, which hopefully will be the day you submit it, but it can be, you can submit it later. And you have to study for at least a half an hour. And if you submit that, then I'll give you an extra credit per week. And so this will be a weekly assignment, an extra credit weekly assignment. And it's just to encourage you guys to get together and study, because when you guys quiz each other, you'll notice that somebody is weak on a topic, and then you'll quiz them further on that topic, and that'll help them. And so it's just something I've set up to encourage you guys to study. All right, any question about any of that? Let me go ahead and turn chapter 5 on. So I'll publish that. Normally I do that at the end of the lesson, but we'll finish the lesson today. And make sure chapter 4 is on. Yeah, it's on. All right. Are there any questions? Or shall we get started with the lesson? No questions. So I believe this was the last slide I discussed yesterday. Yes. Thank you for somebody who ever confirmed it. So we're talking about aerobic respiration, and we've covered glycolysis. If you didn't see that, there's a recording which should be at my YouTube channel. So now we're going to talk a little bit further about aerobic respiration and compare it just briefly with anaerobic respiration, and then I'll finish talking about aerobic respiration, and then we'll talk a little bit about anaerobic respiration and fermentation. Let me blow this up because this part's pretty small. So when we're talking about respiration, what happens is that there's the oxidation of molecules, mainly glucose, which releases electrons, which will be gathered in glycolysis and in the Krebs cycle. And then those electrons are sent to the electron transport chain. There are different electron transport chains between aerobic respiration and anaerobic respiration. In aerobic respiration, the final electron acceptor, meaning the molecule which accepts the electrons, and that's why we're saying it's the final electron acceptor, the final molecule to accept the electrons, is oxygen. And so those electrons flow to oxygen, and then we generate a large amount of ATP from the flow of those electrons in the electron transport chain. Now, in anaerobic respiration, it's the same, meaning it starts with glycolysis, and then we go into the Krebs cycle, and then the electrons flow to the electron transport chain. But the electron transport chain is a little different, and the electrons do not flow to oxygen as the final electron acceptor. They flow to some other molecule that is not oxygen. It could be nitrite or nitrate, or it could be a sulfur compound. It depends on what form of anaerobic respiration the cell is using, what molecule that the cell sends the electrons to. And so there's many different forms of anaerobic respiration, and the final electron acceptor will be different. But there's one thing we can say, and that is the electron will not be oxygen. Okay? And then from those electrons flowing in the electron transport chain, they will generate ATP, and it will be generated by oxidative phosphorylation. So that's just comparing aerobic respiration with anaerobic respiration. Any questions about that? We'll get back to talking about anaerobic respiration after we've talked about aerobic respiration. So in aerobic respiration, after glycolysis, the glucose has been converted into pyruvic acid, and pyruvic acid cannot directly enter the Krebs cycle. In one step, the pyruvic acid is changed into acetyl coenzyme A, and it joins up with coenzyme A, and the acetyl coenzyme A is a 2-carbon molecule. The pyruvic acid is a 1-carbon molecule. That means 1 carbon comes off of pyruvic acid, and it is burned as CO2. That is the first CO2 that we get from the metabolism of what started out as glucose. Now understand that the preparatory step runs twice because glycolysis ends up with 2 molecules of pyruvic acid. So the preparatory step will run twice. And essentially it's converting the pyruvic acid into acetyl coenzyme A. We generate 1 CO2 and we generate 1 NADH. We'll talk more about the NADH that's generated in the preparatory step after we've talked about the Krebs cycle. All right, any question about any of that? I don't have that last part showing. Ah, just a minute. I can't get it to click. Oxygen has not been used yet. So in the preparatory step, oxygen has not been used. So the preparatory step changes the pyruvic acid into acetyl coenzyme A, and now the acetyl coenzyme A can enter the Krebs cycle. Please understand that the Krebs cycle is the term that I'll be using, and is the term that your book mostly uses, but it can be called the tricarboxylic acid cycle, or TCA for short, or it could be called the citric acid cycle, and that's abbreviated TCA also. The Krebs cycle has a lot of tricarbon molecules, why it's called the tricarboxylic acid cycle. And then the first conversion is the acetyl coenzyme A will be converted into citric acid, why it's called that. It's also called the Krebs cycle because it was Dr. Krebs who discovered it, and this is the way I will be talking about it. But it does have different names, and you probably should know them. I won't try to confuse you in a quiz, but other people do call them other things. That's why you should know it. In another class, they'll call it the citric acid cycle or the tricarboxylic acid cycle. All right, here we're seeing the Krebs cycle. Let me go ahead and blow that up. Push that over to the side. My mouse will work. In the preparatory step in the Krebs cycle, so this is actually showing you the preparatory step. The pyruvic acid is turned into acetyl coenzyme A, which can then enter the Krebs cycle. It binds with oxaloacetic acid, and that's converted into citric acid. You don't need to know any of these terms here. The citric acid is converted to another thing, which is converted to another intermediate, and that burns off one of the CO2s, and an NADH is made. And then that's converted into another molecule, another CO2 comes off, and another NADH is made. That's converted to another molecule, and ATP is generated in that step. And then that's converted to another molecule, and FADH2 is generated, which is converted into another molecule, which is converted into another molecule, and NADH is made. And look, we end up where we started. We end up at oxaloacetic acid, which can then join up with acetyl coenzyme A, and then the cycle can start again. And that's why it's called a cycle, because you end up where you start in the cycle. Any questions about that? So in the Krebs cycle, we generate 1 ATP, 4 NADH, and that's counting one of the NADHs we got in the preparatory step. 1 FADH2, I'll point that one out because there's only one of them right here. And then 3 CO2s, one in the preparatory step, one right here, one right there. But the Krebs cycle and the preparatory step runs 2 times because glycolysis generates 2 pyruvic acids. So the second one will go through the preparatory step, and then we'll enter the Krebs cycle, and so we'll run this 2 times for each molecule of glucose. Does everybody see that? So if you're talking about what's produced per molecule of glucose, you just multiply by 2. So for each molecule of glucose, we get 2 ATPs. We get 8 NADH, 2 FADH2s, and 6 CO2s. That means every carbon in glucose, which has 6, is converted into a carbon in CO2. Does everybody see that? And then we generate a fair number of NADH and a few FADH2s, and these molecules carry electrons to the electron transport chain. All right, any questions about the preparatory step and the Krebs cycle? Next, we go to the electron transport chain and the NADH and the FADH from the Krebs cycle, and the NADH from glycolysis carry the electrons to the electron transport chain. In the electron transport chain, the ATP production kicks into high gear. So we can use this slide actually. At this step, there's only been a little bit of ATP made. We made 2 in glycolysis from a molecule of glucose, and we made 2 in the Krebs cycle from a molecule of glucose. So we've only generated 4 so far. Everyone see that? The rest of the ATP will be generated in the electron transport chain of aerobic respiration. The electron transport chain, or ETC for short, is a series of carrier molecules that are in turn reduced and oxidized as electrons flow from glucose, and then they're passed down the chain of molecules in the electron transport chain. And that's why we call it an electron transport chain. The molecules are all together in a membrane. I'm trying to show you a molecule with my hands. And there's just one after another, after another, after another in the membrane. And so there are a chain of molecules, and the electron flows from one molecule, which they're actually proteins, in the chain. So they're transported from one molecule to the next, to the next, to the next in the electron transport chain. And when they're transported, the energy that's released from those electrons can be used by chemiosmosis to produce ATP. Any questions about that? Let's see if we can get this little video to work. I don't know why I have to download the Flash player, because I've got it on here. Here we go. Can you guys hear that? Nobody's answering, so I'm assuming you can't hear it. Let me get this back.
It's slow, but I can hear it.
You can hear it. Let me see if I selected that. Nope, I didn't select that. All right, let's go back. You should be able to hear it now. Do you hear it now? Well,
yes.
So you did hear it?
Yes.
Okay, thank you.
The electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. Electrons captured from donor molecules are transferred through these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex to synthesize ATP. The following complexes are found in the electron transport chain: NADH dehydrogenase, cytochrome VC1, cytochrome oxidase, and the complex that makes ATP, ATP synthase. In addition to these complexes, 2 mobile carriers are also involved: ubiquinone and cytochrome C.
You don't need to know
Other
these
key
names.
components in this process are NADH and the electrons from it, hydrogen ions, molecular oxygen, water, and ADP and PI, which combine to form ATP. At the start of the electron transport chain, 2 electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of 1 hydrogen ion for each electron. Next, the 2 electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. 1 hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate. The ATP synthesis steps you see here are discussed in greater detail in the ATP synthase gradients animation. This animation illustrates 2 full cycles of electron donation. In biological systems, however, many electron transport cycles occur simultaneously, helping to ensure that the proton gradient is always maintained.
All right, any questions about the video? So let's talk a little bit about the electron transport chain. In eukaryotes, it occurs along the inner membrane of the mitochondria. Everybody got that? So this membrane here, which has the electron transport chain, meaning a bunch of proteins are embedded in the membrane. It happens in the inner mitochondrial membrane. For prokaryotes, this occurs on the cell membrane. So it's a membrane, and in eukaryotes, it's the inner mitochondrial membrane, and in prokaryotes, it's the cell membrane. Prokaryotes use the cell membrane because they don't have a mitochondria. And both of them have a bunch of proteins embedded in the membrane, and as I mentioned, that's why it's called the electron transport chain. The electrons are brought to these proteins in the membrane by NADH, which donates the electrons here, or by FADH2, which donates the electrons here. So you'll note that FADH2 brings the electrons later down in the chain than NADH. And so NADH is more efficient at pumping hydrogen ions across the membrane because it donates the electrons at the first transport protein, and FADH2 sends them to the second or third protein. And then when they donate the electrons, these electrons move from one protein in the membrane to the next protein in the membrane. And then to the next, to the next, to the next. And when they're at the last protein in the chain of proteins in the membrane, the electrons then flow to the final electron acceptor, which in aerobic respiration is oxygen. And when the electrons flow to oxygen. Some hydrogen ions come and add to the electrons in oxygen and convert oxygen into water. And that is why we have to breathe in oxygen. All of the oxygen we breathe in is converted into water by the electron transport chain of aerobic respiration. Any question about that? Some of that water is recycled and it joins our blood and it's a water supply for us. Some of it is breathed out. For desert animals, they conserve at least 60% of the water that they get from aerobic respiration, and it's an important source of water for desert animals. Like I said, it accounts for about 60% of their water intake. Let's see, what else do I want to talk about with here? Oh, yes. When the electrons move through some of the proteins in the electron transport chain, for this protein and that protein and that protein, it gives the proteins the energy that they need to pump hydrogen ions from this side of the membrane to that side of the membrane. And that creates a twofold gradient. There will be more hydrogen ions on this side of the membrane than on this side of the membrane. There's also more positive charges on this side of the membrane than on this side of the membrane. And the positive charges are simply because we're pumping hydrogen ions. By the laws of diffusion, this gradient, meaning more hydrogen ions on this side than this side, will want to move to this side of the membrane. Do you guys see that? But hydrogen ions cannot cross a lipid bilayer without assistance, and the only place the hydrogen ions can cross from this side of the membrane to that side of the membrane is through this protein, which is an ATP synthase. And when the hydrogen ions cross through this protein, simply following the laws of diffusion, they give the protein the energy it needs to make ATP from ADP, an inorganic phosphate. And that's how the cell makes most of its ATP. That would be 32 to 34 ATP molecules made by this twofold gradient, meaning this gradient is used, the potential energy in this gradient is used to make ATP. When you have more hydrogen ions on this side than that side, that means there's a membrane difference. What's the word that I'm trying to say? Potential energy difference between this side of the membrane and that side. And that potential energy is used to make ATP. Any questions about that? So I already stated that how much ATP is made per molecule of glucose for the electron transport chain of aerobic respiration is 34 are made in prokaryotic cells. In eukaryotic cells, it's either 32 or 34. Most of the cells of your body can only make 32 molecules of ATP in the electron transport chain. But there are 3 cells which are more efficient, and the more efficient cells are cells of the heart, cells of the liver, and cells of the kidney. And they're more efficient, and so they can generate 34 molecules of ATP per molecule of glucose. Why do you think the cells of the heart are more efficient than the cells of your skin or the cells of your nose? Anyone
Got
have
a
a
lot
guess?
more work to do.
Yeah, the cells of the heart, the liver, and the kidney, they're always working. So they're more efficient at generating ATP. Okay? And in aerobic respiration, once again, remember the final electron acceptor is oxygen, and the oxygen combines with hydrogen ions and those electrons to form water. And that's why we have to breathe in oxygen. And the electrons move or flow from one molecule. Eventually they start from glucose and they're carried by NADH and FADH to the molecules electron transport chain. And these are the molecules of the electron transport chain. You don't need to know these molecules, but they are the molecules of the electron transport chain. And as each molecule accepts an electron, it will be reduced, gaining electron reduction, and then the electron moves to the next molecule, so that molecule will be oxidized. And the same with FADH2 and NADH. So NAD becomes reduced to become NADH, and then NADH is oxidized to become NAD as the electrons flow to the first molecule of the electron transport chain. The point is, as these electrons move down the chain, they lose energy. Some of the energy is used to pump hydrogen ions from one side of the membrane to the next, and then the electrons eventually flow to the final electron acceptor, which is oxygen, and it'll be converted into water. Just showing you where this membrane is. For eukaryotes, it's on the inner mitochondrial membrane. So this is where the electron transport chain is. In prokaryotes, it's on the cell membrane, so it's right here. Okay? And when the electrons are pumped from this side, not the electrons, the hydrogen ions are pumped from this side of the membrane to that side of the membrane. In a eukaryote, the hydrogen ions will be pumped inside the intermembrane space of the mitochondria. So these hydrogen ions are not in contact with the rest of the eukaryotic cell. They're in the intermembrane space of the mitochondria, and that's a good place for them to be because they won't do damage to the rest of the cell. Okay? Any question about that part? In the prokaryotic cell, where are these hydrogen ions pumped to? I'll give you a hint. Here's the cell membrane. Say again?
Cytoplasm?
No, they're pumped outside of the cytoplasm on this side of the cell membrane. Somebody's trying to answer, but I can't hear you. All right, they're not saying again. They're actually pumped into the cell wall of a prokaryotic cell. And then they flow through the ATP synthase back into the cytoplasm. But when they flow through ATP synthase, the energy of the electrons is used to make ATP. Okay? All right. Any questions about this picture? I've already talked about this, and so we'll just mention that the gradient of hydrogen ions on this side of the membrane compared to that side of the membrane can be called the proton motive force. and it's this energy in the proton motive force which is used to make ATP. When the hydrogen ions move from this side of the membrane through ATP synthase to make ATP, this process is called chemiosmosis, the movement of hydrogen ions to make ATP, and it's just a special term for that. Anyways, the energy is used to make ATP. And the process is called oxidative phosphorylation. Phosphorylation is when you add phosphate to a molecule, and we're adding inorganic phosphate to ADP. And then the ADP is oxidative phosphorylated to become ATP. So for a molecule of glucose, it becomes completely oxidized to CO2 via aerobic respiration, and we get 36 to 38 ATP molecules in eukaryotes for each molecule of glucose. And in prokaryotes, we get 38 ATP molecules per molecule of glucose. So the prokaryotes are a little more efficient than most, come on mouse, are a little more efficient than most eukaryotic cells, but there are exceptions, meaning the heart, the liver, and the kidney are more efficient, and they are as efficient as prokaryotic cells. Okay? And so the overall equation summarizing aerobic respiration, which you do need to know, is that glucose, C6H12O6, will combine with 6 oxygen molecules and 38 ADP molecules and 38 inorganic phosphates to be converted into 6 CO2s, 6 water molecules, and 38 ATP molecules. We're talking about for prokaryotic cells here. All right? None of the molecules of oxygen are converted into CO2. All of this CO2 comes from the carbon in glucose. And the glucose is burned into CO2. And then the oxygen is converted into water. Okay? So that's the summary of aerobic respiration. And there you see a summary in the flow diagram, starting with glycolysis, generating 2 ATP, 2 pyruvic acid, 2 NADH. We then go into the preparatory step, which generates 2 CO2 per molecule of glucose, 2 NADH, and the pyruvic acid is converted into acetyl coenzyme A. For each acetyl coenzyme A, we generate, for each molecule of glucose, we generate 4 CO2 in the Krebs cycle with 2 in the preparatory step. That makes 6. So each of the molecules of carbon and glucose is converted into CO2. We generate 2 ATP in the Krebs cycle, 6 NADH in the Krebs cycle, and 2 NADH in the preparatory step, for a total of 8. And then 2 FADH2 in the Krebs cycle. And then these molecules of NADH and FADH2 carry the electrons to the electron transport chain. In prokaryotes, we generate 34 ATP molecules, and we generate, sort of can't see that, see if I can move that over. We generate, we use 6 oxygen molecules, which are converted into 6 water molecules by the electrons and 12 hydrogen ions. You do need to know the numbers of ATP that are generated in each step, as well as what's happening to glucose and how it's converted into pyruvic acid and then acetyl coenzyme A, and then converted into CO2. Okay? So that's it for aerobic respiration. Let's talk briefly about anaerobic respiration. That's another form of respiration. The main difference is in anaerobic respiration that we don't have the electrons flowing to oxygen. It flows to some other molecule. And it depends on what form of anaerobic respiration it is, because each different form of anaerobic respiration has the electrons flow to another molecule. It could be a nitrite or some nitrogen compound. It could be a sulfate ion or some sulfur compound. Like I said, there's different forms of anaerobic respiration, and they use a different final electron acceptor. In anaerobic respiration, we do have glycolysis, the preparatory step, and the Krebs cycle. And glycolysis, the preparatory step, and the Krebs cycle operate the same as they do in aerobic respiration. Any question about any of that? So anaerobic respiration has glycolysis, the preparatory step, and the Krebs cycle, and they operate the same as they do in aerobic respiration. The electron transport chain, though, is a little different in anaerobic respiration from aerobic respiration. The electron transport chain is a little shorter in anaerobic respiration, and it depends on which form of anaerobic respiration we're talking about for how much shorter the electron transport chain is compared to aerobic respiration. So let's end the slide here and then go back to our picture of aerobic respiration. Why aren't I seeing that? Well, that one will do. So as you can see in aerobic respiration, we have a number of proteins in the chain, in the electron transport chain. In anaerobic respiration, we'll have fewer proteins, depending on which form of anaerobic respiration we're talking about. So there will be fewer proteins in anaerobic respiration. Now let's go back to where we were. Consequently, because the electron transport chain is shorter, we will generate fewer ATP because there's less pumping of hydrogen ions across the membrane in anaerobic respiration. Any question about that? The ATP yields differ for each form of anaerobic respiration. The worst, meaning the worst at making ATP, there's anaerobic respiration that only yields about 2 ATP molecules per molecule of glucose, and that's the worst forms of anaerobic respiration. The best forms of anaerobic respiration generate 36 molecules of ATP per molecule of glucose. And so the best form of anaerobic respiration generates fewer ATP than aerobic respiration, which in prokaryotes can generate up to 38 molecules of ATP. Okay? So that means anaerobes tend to grow slower than aerobes because they are generating less ATP in anaerobic respiration than an aerobe is making. And it depends on which form of anaerobic respiration we're talking about. The best are close in generating ATP, close to the number that are made in prokaryotic cells. But as you go in other forms of anaerobic respiration, you'll get less and less ATP generated, and the worst generate only about 2 ATP per molecule of glucose. Any question about that? Okay? So we've talked now about respiration, both anaerobic respiration and aerobic respiration. We talked about aerobic respiration in detail because it's the most efficient way that cells have for making ATP, and it is the way that your cells make ATP. The exception is your muscle cells can make ATP by fermentation, but they do prefer to make ATP by aerobic respiration. All right? So when we talk about how efficient the cell is at making ATP, and we're talking about the efficiency of making ATP by aerobic respiration, the cell is 20% efficient. Is that right? Or is it 40% efficient? Gosh, I can't remember. Anyways, you say that that's not very efficient, converting the energy in glucose to the energy in ATP. Well, the cell is twice as efficient as the best motor that humans have ever made. So when we have a motor that converts gasoline, which are the best motors we have, into energy, meaning the movement of the car, the best we have is about half as efficient as the cell at converting glucose into ATP. So that tells you how efficient cells are at making ATP. About twice as efficient as the best motor that humans have ever made. Any question about that? No? All right. So then let's talk about fermentation. That's the other way that cells can obtain ATP from catabolizing glucose or some other molecule of food. And cells can engage in fermentation, and you should understand that fermentation always starts with glycolysis, and then fermentation has 1 or 2 fermentation steps after glycolysis. Any questions about that? In fermentation, not much ATP is generated. The cell releases the energy from glucose, and the electrons flow from glucose to an organic molecule in fermentation, which starts with pyruvate or pyruvic acid. You get a net gain of 2 ATP molecules, and these ATP molecules made by fermentation come from glycolysis. You do generate NADH and H+ atoms in glycolysis, but in fermentation, the NADH and the hydrogen ions are not kept around and passed to the electron transport chain. They are utilized in the fermentation step to convert the pyruvic acid into some other molecule. And this other molecule will be the last step of the fermentation step. And it'll depend on what fermentation reaction we're talking about. It could be lactic acid, the last molecule made by the fermentation, or it could be ethanol, and it depends on what this molecule is, what fermentation step it is. And there's about 5 or 6 fermentation steps, meaning different fermentations. Fermentation does not require oxygen. It can happen in the presence or absence of oxygen because it doesn't use oxygen. Fermentation does not use the Krebs cycle or the electron transport chain, but it always uses glycolysis. And an organic molecule is used as the final electron acceptor, becoming the end product of fermentation. Okay? Any questions about that? Yeah, let's talk about this one here first. The NADH, which is generated by glycolysis, is used in the fermentation step to make the fermentation end product. And so this NADH is not kept for the electron transport chain. All right? If the cell were to keep this molecule and send it down the electron transport chain, the cell would make more than just 2 ATP molecules per molecule of glucose, because the electron transport chain is very efficient at generating ATP. Why do you think the cell, like your muscle cells, don't keep NADH around and then pass it through the electron transport chain? Why do your cells use the NADH up in a fermentation step, generating no ATP in this step? Doesn't sound to be very efficient. So why does the cell not use that NADH by the electron transport chain? Come on, we're talking about your muscle cells here. Nobody has a guess. Well, if we didn't use up the NADH in the fermentation step, we wouldn't make NAD+. And you need NAD+ for glycolysis to happen. So in fermentation, the NAD+ is being regenerated, so we continue doing the fermentation or continue doing the glycolysis. Because without NAD+, glycolysis won't happen. No glycolysis happens, you don't generate any ATP. And remember, your muscle cell is only doing fermentation when oxygen is limited, so the electron transport chain cannot function because there isn't oxygen around for your muscle cell to perform the electron transport chain. And that's why your muscle cell engages in fermentation, because oxygen is limited, it can't perform the electron transport chain. The muscle cell still wants ATP so the muscle can work, and so it goes through fermentation to generate some ATP. And it's not very much. It's only 2 molecules of ATP for each molecule of glucose. But it is enough for the muscle to keep working. Okay? And the heart and the lungs are working as rapidly as they can to bring oxygen to the muscle. It's just the muscle is working so strenuously that it is using up all the oxygen that is brought in aerobic respiration, and then the muscle needs to keep working, and so it engages in fermentation to generate some ATP. Now, what happens to an Olympic athlete when there's no longer glucose available, meaning all the glucose in the muscles are burned up? What happens to the Olympic athlete when there's no more glucose available? And obviously, the Olympic athlete is running towards the finish line or doing whatever they are, getting close to the finish line, and they're engaging in fermentation in their muscles because they've been pushing themselves to the limit.
Your muscles cramp.
They don't cramp. Anyone else have another guess? Cramping is when the muscle is working and what do you call it? Is contracting, and that takes ATP to contract. Okay?
Is it a buildup of lactic acid?
Yeah, that happens, but that happens every time even those of us who aren't Olympic athletes go running or something like that. Lactic acid is building up, yes, but what happens to the Olympic athlete when they run out of glucose in the muscle cell? They can't engage in fermentation.
Muscles feel sore.
Their muscles are sore, yes, because of the buildup of lactic acid, but that'll happen even when there's glucose available and they're engaging in fermentation. No, I'm saying what happens to their muscles when they can't engage in fermentation because they have no more glucose left. You usually see it about once in the Olympics. One athlete is getting close to the finish line and they're engaging in fermentation because they're pushing their muscles to the max, and then they
collapse.
Yeah, they collapse. They have no more ATP generated. Their muscles just stop working. They collapse. Okay? Their coach comes out running, and if it's the Winter Olympic, they throw a blanket over them. I assume in the Summer Olympics, they just spray down the Olympic athlete. And then the heart, the heart and the muscle is working like mad to bring oxygen and glucose to the muscle. So it'll come to the Olympic athlete. They'll get up and then hobble across the finish line. Okay? Meaning they're fine. It's just that their muscles stop working because they don't generate any more ATP. And you usually see that about once in the Olympics. And some of you actually remembered that. Good job. All right, let's talk about the different fermentation reactions. There's 5 or 6 of them. I'll show you a slide of it. And there's only 2 that you need to know. Lactic acid fermentation. A lot of bacteria can perform lactic acid fermentation, and we use that to make yogurt, to make chocolate, to make, let me think of what else, sauerkraut, to make sausage, to make a lot of products. Most cheese is produced by lactic acid fermentation, by either a bacteria or a mold. Okay? So it's very common. A lot of organisms engage in lactic acid fermentation, including your muscle cells. When they are short on oxygen, they perform lactic acid fermentation to make ATP. And most of us aren't Olympic athletes, so we never have to worry about running out of glucose in the muscles and then we collapse. Okay? I've never had a student say that it happened, but it doesn't happen very often. And usually you only see it in the Olympic athletes. And even among them, in the entire Olympics, it's usually only about once in the Olympics that it ever happens. In fermentation, you convert glucose by glycolysis to pyruvic acid, and you generate 2 ATP. That's all you generate in fermentation. Fermentation with lactic acid fermentation has one more step. Let me blow that up because it's kind of small. Fermentation, lactic acid fermentation is shown right here. In one step, the pyruvic acid is converted to lactic acid in the lactic acid fermentation. All right? Any question about any of that? Trying to move that over. So lactic acid starts with glycolysis, and fermentation always does, and then the pyruvic acid is converted into lactic acid in one step, one fermentation reaction. And we use that to prevent food from spoilage. Like sauerkraut can be kept at least a day at room temperature. I'm not sure how long you can keep it at room temperature, but in the refrigerator you can keep that close to, oops, sorry I advanced, close to a year. Okay? Yogurt, you can keep it at least a day at room temperature, and I don't know how long you can keep it in the refrigerator. A long time, more than a week, meaning it has lactic acid in it and it prevents the yogurt from spoiling. Another fermentation reaction that you should know is alcoholic fermentation, also called ethanolic fermentation. This takes two steps. The pyruvate is converted to an intermediate. You don't need to know the name of it. CO2 is given off, and then that intermediate is converted to ethanol. Ethanol is a 2-carbon molecule. Whoop, right there and right there. And pyruvic acid is a 3-carbon molecule, and that's why carbon dioxide is generated. The carbon dioxide is the fizz in the beer you taste, or in wine, it's created in a barrel or someplace open, and so the wine, the CO2 just enters the atmosphere. But if you were to cork the bottle of wine, it would be fizzy, and we call that champagne. Okay? And that's ethanolic fermentation, also called alcoholic fermentation, and it takes 2 fermentation reactions in alcoholic fermentation. You are familiar with that even if you don't drink beer or wine, in that this is also the fermentation reaction you have when you have yeast and you make leavened bread with yeast. So that's alcoholic fermentation, and that would be sourdough bread. Okay? If you only are using natural yeast, it'll make sourdough bread. This is the reaction that happens to make leavened bread if you add the package of yeast and then you add the sugar. The yeast converts some of the glucose or the sugar to ethanol, and when you're baking it, that ethanol bakes out of the bread. Why you don't taste it in when you make the bread. Okay, you have to make sourdough bread to actually taste the ethanol, and you don't taste ethanol because the sourdough makes an acid as well as the ethanol. All right, so those are the 2 fermentation reactions you need to know. There are different fermentation reactions: lactic acid fermentation, ethanolic fermentation we discussed. There's propionic acid fermentation, and this is the fermentation that is used to make Swiss cheese. Swiss cheese is different from almost all other cheeses which use lactic acid fermentation, like cheddar cheese and American cheese and cottage cheese and almost any other cheese you can think of. Okay? A Swiss cheese uses a different fermentation reaction, and it's propionic acid. Humans also use butyric acid fermentation to make butanol, acetone, and isopropyl alcohol. Those have industrial uses. Escherichia, meaning like E. coli and Salmonella, can have mixed acid fermentation, making lactic acid, succinic acid, acetic acid, and ethanol. Enterobacter, I think this is the one that can make formic acid, which is of vinegar. And we can also make other products out of that. All right? So those are the different fermentation reactions. Like I stated, you only need to know these two. The fermentation reactions have a lot of industrial uses because humans use the different fermentation reaction to make different products, like isopropyl alcohol. And this is showing you some of the different products that are made by fermentation. So actually vitamin C, I didn't know that, was made by a fermentation reaction. You can also get vitamin C naturally in fruit. Fuel can be made from fermentation, and all of these products here. Okay? You don't need to know this table. It's just showing you the different fermentation reactions. And the point is, a lot of the food we make is made by fermentation. So this table is giving you a good summary of aerobic respiration, anaerobic respiration, and fermentation. In aerobic respiration, you need oxygen for aerobic respiration. You do not need oxygen for anaerobic respiration or fermentation because the final electron acceptor is not oxygen. It's some other molecule, and that's all you need to know about what molecule it is for anaerobic respiration. And in fermentation, you just need to know it's another organic molecule of the fermentation. I won't ask you about the type of phosphorylation used to generate ATP, but you should know this column, the ATP that are generated per molecule of glucose. It's 36 to 38 in eukaryotes. Most eukaryotic cells generate only 36 ATP per molecule of glucose, but prokaryotes generate 38 ATP molecules per molecule of glucose. And anaerobic respiration, the ATP produced is variable, but it's between 2 and 36, depending on what form of anaerobic respiration we're talking about. And then fermentation generates only 2. Any questions about this slide? All right. So apart from glucose and the other carbohydrates which will be converted into glucose, your cell can eat like bread and convert the bread into glucose and then undergo aerobic respiration in the normal way using glucose. You can also use lipid and protein catabolism in aerobic respiration. So other foods can be used, and microbes can do this as well as you. There is a problem, and that is large lipids and large proteins are not going to cross the cell membrane. But a microorganism has a simple solution, and actually that happens in your gut as well. Large lipids and large proteins cannot cross the cell membrane and into the cell. So we digest these molecules outside of the cell. We make enzymes and produce them in our gut and then digest the large lipids and the proteins, making them smaller monomers. And then we bring the monomers inside the cell. Well, a microorganism does the same thing, except they don't have a gut. They make the enzymes like proteinases and lipases, put them outside the cell, digest the lipids and the proteins, convert them into monomers, and then bring the monomers across their cell membrane. And then engage in aerobic respiration, since we're talking about that, inside the cell. And they use lipids and proteases. And then with using the monomers, they go through the normal steps of glycolysis, the preparatory step, the Krebs cycle, and the electron transport chain. It is true with the digestion of proteins that in an amino acid, for it to go through aerobic respiration, the amino group has to be pulled off of the amino acid, and then that amino group has to be removed. And in organisms, that will be converted into urea, I should say in animals, that will be converted into urea and then excreted out. Prokaryotes do something similar. It's just that they don't pee. They convert it into urea and then dump it outside the cell. Let me show you this in a little blown up form because it's kind of small. So let's talk about lipids because they're a little easier to talk about. All carbohydrates will be converted into sugar and then go through aerobic respiration in the normal way. Any question about the way carbohydrates are digested by aerobic respiration? So they'll be just mainly converted into glucose and then go through aerobic respiration in the normal way. Lipids are not worked that way because they aren't a carbohydrate. They will be broken down into fatty acids and glycerol. Glycerol can join the Krebs cycle midway through, and then that'll go through in the normal way of aerobic respiration. The fatty acids are broken into 2 carbon chains and then converted into acetyl coenzyme A. So the fatty acids join aerobic respiration as acetyl coenzyme A. Okay? Any question about how lipids join aerobic respiration? Proteins are complex because there's 20 different amino acids. All of the amino acids will be converted into a carbohydrate-like molecule, meaning the amino group will be pulled off, and that's not shown in this slide. And in animal cells, the amino group will be converted into urea, and then that'll go into the bladder. Anyways, the amino acid, for some amino acids, will be converted into pyruvic acid and then go through aerobic respiration that way from that point in the Krebs cycle. Some amino acids will be converted into acetyl coenzyme A and then join the Krebs cycle. And some amino acids will join the Krebs cycle way down, meaning near the end of the Krebs cycle. Okay? Depending on what amino acid we're talking about. Any question about how proteins can be digested and enter the aerobic respiration cycle?
It's 2:15.
Oh, okay. All right, let's go ahead and stop this, and we're almost done. We'll finish it in the later lab. Thank you. Any questions? All right, I'll see you